notice that you don't see, usually the most familiar thing that people know about DNA is when they see it looking like this, where it has a particular shape. And here you don't see that shape. You just see all kind of fuzzy stuff that's spread out throughout that nucleus. And so that taps into the use of the word chromatin. It has a special meaning. It means that the cell is not actively dividing, and it's not actively undergoing mitosis. When it is not in the process of getting ready to undergo cell division or mitosis, it is not what's called supercoiled. It's kind of in a relaxed state, so your double helix is kind of spread out all over the cell. And so the name that's given to that is chromatin. When the cell begins to get ready to undergo cell division, which is, for our purposes, mitosis, then you're going to have to start replicating the DNA so that you can get one cell dividing into two to make two daughter cells, and then it gets, that supercoiling is chromosomal. And so, for a quick definition, chromatin is loosely folded DNA. And I'll talk about what else is in there. Plus, and chromosomes refer to replicated, supercoiled DNA. So the plus, here they're showing you the levels of organization of DNA that's inside the nucleus. And so we have talked already about the fact that DNA is a nucleic acid. It's part of the nucleic acid family of organic molecules. Yep.
It's supercoiled.
Supercoiled DNA. And then what happens is, remember, the DNA has the genes in it. And that DNA, the nucleic acid string, is fairly long. And so we want to be able to have the genes accessed to be able to make protein. So if you think of the analogy of putting a ball of string in a drawer and just throwing it in the drawer, and now trying to untangle a certain area of it, if it had a gene there. Probably it'd be all knotted up and ruined. And so the same analogy is happening in the nucleus. You want it to, the best thing to do with the ball of string is to coil it up very neatly. Yes?
What's it say under chromosome?
Loosely folded, plus. And so we're getting into what the plus is. And so the idea is you want to keep this, this DNA that has all these precious genes in it that are going to determine all the so-called traits. Some of the traits are defined as different proteins and enzymes that are in your body. And so the best way to keep it neat is to coil it around. If you have string, you coil it around a piece of cardboard and keep it neat that way. And so the way the chromatin gets folded is to coil it around a protein. And the protein that the DNA gets folded around is called histones. And so these are histone proteins. And so they, under the microscope, they begin to give what's called a bead on a string appearance. So each one of these little yellow spheres is the histone protein, and you get this constant linking. Even though so now it's neat, it's in the drawer neatly. But it is, if the cell is not in the process of dividing, it's going to be loosely coiled. And so if it's loosely coiled, then it gets that name chromatin. And here you can see the supercoiling, or it's called condensation. The whole part of the DNA is replicating, and it's the nucleic acid is tightening up, and it's becoming exactly what they're calling it, a supercoiled DNA. The idea then is going to have a shape. And so here we have the typical shape that we see in textbooks when they talk about chromosomes. So the chromosome is replicated, meaning it has doubled itself. And the cell now actually has 2 times the amount of DNA that it needs. But that's exactly what it needs because it's going to get ready to divide. And so that's the difference in the terminology. And so when they label this as chromatin in the diagram, they're telling you that it is, the cell is in its resting stage. That resting stage is not a good terminology. The cell is still doing a lot of stuff. It's engaging in metabolism, but it's not involved in division at this moment. If you're going on to 106, this is all important information that you should learn now, because the first half of 106 is all the genetics. You're not going to leave that nucleus and that DNA for almost half the semester. And most of the labs are going to be dealing with DNA and replicating DNA and analyzing DNA on PCR machines, etc. Now's the time to just get the basic information. So this is what's in the nucleus. Notice also we said that the nucleus has the nuclear envelope, and we make a big deal about the nuclear envelope. I started talking about it last time, that things have to move back and forth across the nuclear pores, because the DNA is specifying the making of what's called messenger RNA. The messenger RNA then leaves to go out to make a protein. The other importance of here's our sequence of events, and so here is our gene to protein. We don't need the terminology right now. We'll get the terminology, the exact terminology later. But here's your nucleus. Here's your double helix of DNA. There will be some gene on here that specifies the making of another nucleic acid called messenger RNA. So we have different kinds of RNA. M stands for messenger. The messenger RNA, notice, is different from the DNA because it's not a double helix. It's only a single string of nucleotides. That messenger RNA carries the genetic information. So the DNA cannot leave the nucleus. It has to stay. So literally, it's going to send a messenger to go outside of the nucleus into the cytosol to make the protein. And so here, the messenger RNA leaves through the nuclear pores, and we want this to make a protein, so we're going to have it sit on a ribosome. So I think we briefly introduced that organelle last time. A ribosome is the place where proteins are made, and so that taps into the chemistry that we've had previously. Proteins are made from amino acids, and they're tied together by special covalent bonds called peptide bonds. And so all of this is happening from gene to messenger RNA. So you want to do a flow chart. DNA, nuclear DNA, to messenger RNA. And then we go out into the cytosol, where the ribosome is, and with the help of the ribosome. And with the help of the several factors that we're not going to discuss in detail right now, amino acids will join in peptide bonds to become protein. Then they're going to go off into other organelles, because once a peptide, remember, a peptide bond is just a special class of covalent bonds. And so once the peptide, the polypeptide is formed, it has to come into a particular shape. And if it's not its correct shape, then it's not going to function. I think we've already talked about that in the lab. Everything deals with shape. That's why we had the second lab dealing with the molecular modeling lab. And so what's going to happen now is that polypeptide is going to go off into other organelles, and they're going to be given time to fold into their correct shape. They might have other molecules attached to them, and we'll talk about that in a minute. This whole system of a nuclear envelope, and notice it's attached, that polypeptide is going to go off into this structure, endoplasmic reticulum, and then it may go off into this structure called the Golgi complex. And so between the endoplasmic reticulum and the Golgi complex, that's where we can say that the polypeptide was processed. So we can just give a general description of processing to notice that this is a series of folded membranes, this is a series of folded membranes. The whole system, starting from the nuclear envelope all the way out to the Golgi complex, is called an endomembrane system. This folding, remember, is increasing surface area. And so we had talked last time about one of the ways that the cell gets over that problem of volume and being too big and having a greater need for metabolism is to fold, and it becomes much more efficient. So everything that's inside the membrane has been compartmentalized with that folding that serves to increase surface area. The lysosomes are another membrane-bound organelle, and that membrane-bound organelle has in it, digestive enzymes. And so notice where the lysosomes are coming from. The lysosomes are coming from the Golgi complex. And so here are little vesicles, membrane-bound sacs, and so those would be lysosomes. And what are the lysosomes doing? A digestive enzyme. So lysosomes contain digestive enzymes. And a digestive, another word for digestive enzyme is a hydrolytic enzyme. I'm going to put ACEs because anything that ends in ASE is an enzyme. Hydrolytic enzymes mean that they have the ability to break bonds. So if you're breaking a covalent bond, you're breaking something down. And that's what lysosomes are doing. If you eat fats, and your digestive system breaks the fats down into fatty acids. We built fatty acid molecules in lab, huge long chains of even-numbered hydrogens and carbons, or even-numbered carbons with hydrogens, a big hydrocarbon tail. Those have to get broken down into their atoms. The body can't use that big molecule. And so who's breaking it down? It's going to be the lysosomes. The lysosomes get incorporated into the fluid of the lysosome. The hydrolytic enzymes are working in a highly acidic environment and about pH 3 or 4. And the enzymes with the acids break the bonds of those food molecules. If you have toxic waste that has been produced by the cell as a result of its metabolism, that can get incorporated into the lysosome. And the lysosome will do the same thing. The hydrolytic enzymes are released and activated by the acids, and they start breaking down the toxic waste. And so the lysosomes, in order to keep the cell healthy, have to be functioning, and they're going to be made back in here in that Golgi apparatus. And so all of these little blips coming out here are, some of them are lysosomes. This one is probably the lysosome in purple. Here's another membrane-bound organelle called a vacuole. These are, we had talked about this earlier in last class, about the ability of the plant cell to increase surface area by just carving out the whole center of the cell and filling it with a sac of water. And so the central vacuole is a membrane-bound sac filled with water that goes from the root all the way up to the stems and through, well, to be distributed to the leaves. And we said last time that's why plants can get so tall, because they're, if they had to depend on just diffusion of water from their roots, a 60-foot tree would probably not be able to get enough water quick enough. And so here's another vacuole that's in animals, food vacuole. Once again, you're eating, this goes back to the example I just gave, similar amoeba, single-cell organisms that live in the water, have the ability to engulf their food, and then the food will get surrounded by a membrane, and enzymes will come into that food vacuole and digest the food down. So it's similar to the action of a lysosome. Contractile vacuole is another membrane-bound sac that's in a cell. And so I don't have a picture of a contractile vacuole. Contractile vacuole is usually in animals that are living in fresh water. We talked in lab about the problem of hypotonic solutions and what happens to cells in hypotonic solutions. And so you have this little single cell amoeba sitting in fresh water. They have the equivalent of living in a hypotonic solution. They're in danger of having water brought into their cell because they have a higher concentration of sodium chloride inside the cell than out. And so they're in danger of blowing up. And so since they do lysing, we can use the $10 word, they undergo lysis. And so what do they do to adapt? They can't leave the fresh water, they don't have legs. And so they have over millions of years adapted a contractile vacuole. It's kind of like a bilge pump. And so as the water goes into the cell, this contractile vacuole squeezes and just pushes the water under pressure right back out. And so that contractile vacuole is how they are able to survive in a hypotonic freshwater stream, for example. Here's our mitochondrion. We will be talking more about the mitochondria when we get into the medical metabolism chapter, and we have a whole chapter coming up on cellular respiration. And all the ATP that's formed in the cell is made in the most of it is made in the mitochondria, and so your task for this chapter is just to learn the basic anatomy of a mitochondria. One mitochondrion is a double membrane organelle. It has an outer membrane, and then it has an inner membrane. Then it has in between the two membranes an intermembrane space. And then it has inside the inner membrane, it has a consistency that's somewhat similar to cytosol, except that it's called a matrix. So these are all the components of a mitochondrion. The fluid inside the inner membrane is called a matrix. Notice that the inner membrane is folded. Once again, we have that strategy of folding to increase surface area. And that membrane gets a special name. It's called the crista. And then we have the intermembrane space. And then we have the outer membrane. Each one of those terms that I'm using that are identifying parts of the mitochondrion are extremely important because the ability to make ATP, the maximum ATP, is going to depend on what's going on in the intermembrane space. We won't even talk about what it is right now, but we have we're going to have events happening in the intermembrane space that allows the maximum number of ATP to be made. We also have enzymes, complexes that are embedded within the inner membrane, the crista, and those also allow for the maximum number of ATP to be made. And so there's a whole host of genetic diseases that those enzyme complexes that are embedded within the inner membrane aren't made properly, and the organism either lives a life that's very compromised in terms of having energy, they don't have a lot of energy, or they can't survive. And inside the matrix, we have another system going on that we'll talk about in later chapters, where more ATP is made. So a few ATP are made in the matrix. The bulk of the ATP is made within the inner membrane, the crista, the enzymes that are embedded on there, and what's going on in the pumps that are going on in the intermembrane space. So for now, just confine yourself. Don't worry about all those events. You have a whole cellular respiration chapter coming up. Now just worry about the anatomy of the mitochondria in order to know what is placed where. Here's another organelle that's in plants that looks suspiciously like a mitochondria, but it's called the chloroplast. And the chloroplast is the equivalent. For our cells, ATP is the so-called energy organelle. It's the place where most of the ATP, which is our energy molecule, is made. And the chloroplast is the place where the cell's food is made. The chloroplast has the ability to make the cell's sugars that it needs to survive. It also makes ATP. The real product for a plant, the chloroplast, is to make sugars. survival system. And so that's why we can't get rid of plants. So the chloroplast is kind of the analog of the mitochondria in a plant. So it is making some ATP, but primarily it's going to be making the plants with food, usually in the form of polysaccharides. Here is our cytoskeletal fibers. These are other structures. These are proteins. And you have a whole table of them. There are three major cytoskeletal fibers that are in cells. One are called microtubules. These are relatively large, thick protein filaments. And one of the places where they play an important role is in mitosis. And so I have a picture of that. Here's a cell undergoing mitosis. Here's our duplicated chromosome. These are microtubules. And what's going to happen is when a cell is undergoing mitosis and it's dividing, it has to be getting half of the replicated DNA into one cell and the other half of the replicated DNA into another cell. And so who's going to pull it into the proper place but these microtubules? And those microtubules get a special name when we're talking about dividing, and they are called the spindle fibers. And so the spindle fibers are microtubules. You can see the importance of that, or I'll show you the importance of that. Let's say we have a chromosome. I'm going to make it obvious here that this is a duplicated DNA. So instead of drawing a double helix, this one line is going to represent the double helix. The DNA has replicated itself within a cell, and so here's the identical strand of DNA that has been duplicated. So again, I'm eliminating the double helix, and I'm representing the whole DNA strand as a single line. When a cell divides, it has a real challenge. It has to make sure that when it divides, this whole strand here goes into one cell, and this whole strand here goes into another cell. If that doesn't happen, and it doesn't happen in exactly that way, then you've mixed up the genes. So we can make a cartoon out of this. Here would be the head, here would be the tail, or make it a human, make it a foot. This would be the head, this would be the foot. So that means the cell has to make a couple decisions. It has to figure out which way it's going to divide. If it starts dividing this way, we end up with two heads. So it has to divide this way. And so the division of the cell is going to come in like this. Who's making that decision? Part of it is the spindle fibers. So the spindle fibers, in a way that we still don't understand, start to come out from an area in the cell, and they attach to the chromosome this way. And then those microtubules that are called spindle fibers have the ability to contract and shorten. And so as the cell is pinching in this way, it's pulling this whole genetic material into one cell, so you get a head and a foot, and it's pulling the genetic material of this chromosome into this half of the chromosome into this cell, we get a head and a foot. That means that these spindle fibers are the determiners of mitosis, of whether things go right or wrong. Yep.
Can it make a mistake? Like,
Yes, they do make a mistake. And sometimes, well, I can give the example by when scientists or medical doctors deliberately make a mistake. One of the first treatments for cancer, we talked a little bit about cancer. We said that cancer was differentiation, was mitosis without differentiation. The cells just divide and divide and divide, but they never differentiate. And that becomes the tumor that eventually kills the organism. One of the first treatments for cancer was to administer a drug that does not allow the spindle fibers to orient in the proper way and attach to the chromosomes properly. So they would come out from their parts cell, and they would just randomly attach anywhere. We have just a big bundle like that big wad of string we were talking about that's in no particular order. So that when the cell divided, it would have maybe 3 heads and a foot. So the genes were all randomly mixed up. So what happens, of course, the cells don't survive. And so the so-called daughter cells die off. And so that's what happens. And so those drugs were specifically made or discovered in order to disrupt the organization of the spindle fibers. And so, yes, there can be genetic mutations, but the result would be the cell wouldn't survive. And if enough cells have their dividing cells, spindle fibers disrupted, then the organism doesn't survive. So that's the importance of the microtubules. Microfilaments, one of the major microfilaments that we know about is called actin. Actin is a contractile protein. It can shorten and it can lengthen. And so where you're going to talk more about actin is when you get into the skeletal, the muscular system in 106, and you talk about the whole mechanism of how muscles contract. And so in order to have a muscle contract and allow you to move your body, for example, you have to have these actin protein filament shortening. And so that's one importance of those microfilaments. The names microtubule, microfilament, intermediate filament was based on their diameter. Intermediate filaments are intermediate between others. The intermediate filaments, the one example that I'm going to hone on, you can read down here all the places that there are, but the one I'm going to emphasize, intermediate filaments are a huge family of proteins. Part of some intermediate filaments are in your skin. The fact that you can go out in the rain and not have the water soak into the depths of your body is because you have kind of a waterproofing in your skin, and your skin, you control your skin and it doesn't fly apart, and all of that is because of some particular kinds of intermediate filaments. Another kind of intermediate filament is called laminin. And so it sounds like not a big deal. Laminin is a type of protein. There's different families of laminin. One of the laminin families are right on the inside of that nuclear envelope. And so before we had drawn a cell, and here we had the plasma membrane, we had the nucleus, and I just labeled this nucleus, and then I labeled it nuclear envelope, and that's all I put. Envelope. But in reality, what's inside this nucleus is a series of laminin proteins that are lining around here. What's the big deal? And they come out and they kind of go across the nucleus like that. What's the big deal about them? Well, they stabilize the shape of the nucleus. So they're nucleus stabilizers. What's the big deal about that? We just talked about the gene to protein trip. Genes determine the messenger RNA that has to leave through the nuclear pore. Then that messenger RNA goes out into a ribosome, and that ribosome makes a protein. The protein could be anything that the cell needs for survival, or it can be an enzyme that the cell needs for survival. And so that nucleus has to maintain a proper shape. If you have the shape being distorted, then the size of the nuclear pores begin to change. They kind of collapse down on themselves. The whole nucleus can collapse and start destroying the DNA that's inside. And what is the result of that? Here is a disease. It's extremely rare. There's only about 100, maybe 50 to 100 individuals in the entire planet that have the disease. But you can see what happens. It's called progeria. There's different kinds of progeria. Progeria in general is a disease of accelerated aging. And this particular kind is called Hutchinson-Gilford after the individual that discovered it. What's happening in this disease, you can see here, this is a child maybe 5 or 6, and this is the child that's 13. He looks like a man that's in his 80s. You can see the skin is wrinkled, his eyesight has already gone bad, he already has cardiovascular disease, everything that is associated with aging. He has arthritis. And so this is a very bizarre disease of accelerated aging. The cause of the disease has been determined to be a disruption in the laminin proteins. So this child was born with laminin proteins that were not normal, reasons we don't understand. For that reason, all that metabolism, that gene to protein communication that went on was not being done correctly. So the metabolism of every cell in the body was not going on properly, and the result is the cell begins to die. And so these children usually don't live beyond 13. 13 is the average age. That would be equivalent to about an 85-year-old male or female, males and females who are affected by progeria. And so this particular kind of progeria is one of the outcomes of not having that laminin properly regulated or structured within the inside of the anterior endpoint. Yep.
So, if there's no interference to shape, from this morning?
Yeah, it kind of, it becomes misshapen, and everything that is associated with the nucleus being misshapen, the chromatin is no longer working properly because it's probably not folded properly around the histones. That means when genes start to be activated, they are not activated properly because they don't have access to, they're not in the proper environment to make the messenger RNA. And so it all starts at the genetic level. And it starts at the genetic level because the laminin is not holding. It's like a house. If the house collapses on you because it's misshapen or there's weak struts or studs in the house, it's going to crush you. And so essentially that's what happens with this laminin. It allows the nucleus to just kind of implode in on itself, damaging all the DNA and genetic controls that are going on within the nucleus. There's another kind of accelerated aging situation that is also involving improper nuclear stabilization. This one is called Werner syndrome. And so this is a little bit less bizarre than progeria. One of the interesting things about progeria is all these children look alike. Genes typically, there's very few genes. There are point mutations where only one thing is affected, like hemoglobin, where only one amino acid out of 244 is affected. But often when you have a genetic mutation, it'll affect a lot of traits because genes are often interlinked. And so these children, if you look carefully, they all look very similar. Their head is usually too large for their body. Their ears are usually disproportionately large. They have a recessed chin. Their nose is usually beaked, what's called a beak, very small and sharp. Their eyes are usually disproportionately big, and they're all aging at a very rapid rate. And so that obviously means that multiple genes are affected. So this is the most bizarre and the most disastrous case of progeria. Werner syndrome, you're still accelerating at a rapid rate, but notice here's the patient at 48. So these people live to be much older than 13 years that the other progeria. So here, and this is the same problem. It's a problem in the nuclear stabilization. The nucleus has not been able to maintain its shape, and everything is going on inside the nucleus. So here is a patient at 15 years old, 48. She's looking more like 80. She already has muscle damage. She already has the thinning of the hair. She already is losing kind of a wasting disease situation. And this is ethnically related. A lot of people of Asian descent, not a lot of people, but when you see Werner syndrome, it's of people of Asian descent. So again, we have genetics in terms of different cultures. So these are two syndromes, very rare. Again, progeria, the other kind of Hutchinson-Gilford progeria is maybe 50 to 100, no more than 100 people in the entire documented case of history have known it. but it's bizarre, it's the most bizarre, it's studied because people have always been looking for the secret to aging, why do we age? We have all these theories, some of which we've already talked about, the membrane, cholesterol inserts, rigidity, etc. And now we're looking at aging at the level of DNA and nucleus. And so, as a medical aspect, as a scientific endeavor, these patients are of extreme interest to people because it kind of holds the key to aging. Okay, so that's, if I haven't convinced you of the importance of those laminins and those filaments that we're talking about, those protein filaments. Here is actin. In more detail, we said actin was a motor, actin and myosin contract the muscle, and with myosin being what's called the motor protein, and then actin is the filament that we were just talking about, the microfilament. And so when you are in 106 and you're talking about muscle contraction, you're going to come back to the relationship between this actin and myosin. And so actin is prominent in the entire muscular system. Myosin is in the purple, and so when they say it's a motor protein, this little knob of protein up here has the ability to change its shape up and down. And so this would be actin here, and myosin can do this. So it can move the actin towards the center, and as you'll learn, that is the muscle contraction. Muscle, when it shortens, the actin is actually moving in towards that empty space. So you have a myosin-actin interaction. So actin is everywhere. Actin is in any of that contractile vacuole that we were just talking about, the bilge pump, being able to have animals that are in hypotonic solutions, being able to push the water out. Those contractions of that bilge pump is due to actin. Here is the contractile. Here's the contractile vacuole here. Okay, that's it for the cell. Again, the hallmark of college education is taking good notes. So if you have taken notes, I have not gone over everything that's in the chapter. And so make sure that you read accordingly. The idea of coming to class, you take the notes, and then you go back into the chapter and fill in and expand what I might have glossed over or quickly went over or something that you missed. And that's where the notes become a useful study tool. It also is a guide, because that means you know what I emphasize in class and what you don't have to spend hours on. If you don't do that, then you probably won't be discussing actin myosin, because you won't be in 106. Okay, we're not done. We have the next chapter coming up, which is metabolism.